So let us see multiple ligand docking with software called pyrx. Okay, so you double click on pyrx. So it is for multiple ligand docking. It takes few seconds. The interface will appear like this. So here you can dock multiple ligands. Okay. So here what you do, first I will create a folder. So I will create a folder and I will rename it as pyrx. Okay. So I am giving it as P Y R X. So we are going to work it as pyrx. Already contains pyrx. Okay. So we will rename this. We will delete this and we will rename this. P Y R X. Okay. So I have created a folder. So look into this folder. There is nothing in this folder. Okay. So here what you have to do, first you have to set the preferences. Similarly what we have done in the Autodock. So file, go to, you have to go to edit and preferences. Go to edit and preferences. There the preferences in the file tab. Okay, here. So you have to create you other things you leave it. You go to the workspace. So workspace you browse and you select pyrx and you click OK and you click OK. So after setting this preference, you just close this. Okay, first time you have to do this. Any work if you start, you have to go this again. So if you see the pyrx, there is nothing inside. Then again, if you go there in the softwares, again we'll open pyrx. Okay. So when you open this, now you can start working on this. So you can see, we will minimize and we will see inside pyrx. So it has created some folders. So now the working directory is ready. Okay. So here you can keep the ligands, results, macromolecules and all you can keep here. See from the Autodock, I am going to take the protein. Okay. So you have the naked protein which is processed with Swiss PDB. So I am taking that, copying that. So I have the protein in my hand. Okay. So then I already told you how to uh, know the ligands. So in the introduction also I have given in the ligands. So what are the ligands which are taken from this? So since I have already downloaded this, so we will take those ligands. Okay. We will close the introduction and then we will go to the get the ligands. I already saved this. So we will go for pyrx ligands. So I will go to select all the ligands. And then I am going to place here. Okay, I have all the ligands. I am going to dock this multiple ligands. And then I am going to use this. Okay. So this ligand, whatever I have meant here is I have downloaded the Zen Aristat from the PubChem. Okay. Suppose if you download in, for example, from the Chimera, you can interconvert it using Open Babel. Okay. So first we have to cluster these ligands. Okay. So how we can do? You have to open BioVia Discovery Studio. Okay. So Discovery Studio you have to open. Okay. So here you go to File and you have to New Molecule window. First you have to click this. Then file, you go to insert from file. So first you have to go to file, new molecule window. Then file, insert from file. So we have already created the working directory as pyrx. Okay. So in the pyrx, so we have all the ligands. You go to select all the ligands. You can select individually or you can press shift button and select all the ligands. Okay. Then you open. So you can see all the ligands will get opened in this. So you select all the ligands. One. So like this, you can cluster all these ligands. Okay, we have clustered these ligands. So you can see this. So go to file, then save as. Okay. So you have to save it as a single cluster. So you have to save in SDF format. So I am giving it as ligands underscore cluster dot SDF. So you have to save it. So we have clustered all the ligands. If you have 100 or 1000 ligands, you have to do this step. So all these things you have to cluster. So it is the chemical information ID. So then you can close this. So in the pyrx folder, we have the ligands underscore cluster. Okay. So now what you do, so you go and file, import. So go to file and import. You create chemical table file SDF. You click next. So we know the directory. So in the desktop, you have the pyrx folder. So in that, you have the ligand cluster. So ligands underscore cluster. Okay. So it has taken all the ligands. You can see the molecular weight, number of atoms and all. So here, what you have to do, you go and right click. So when you do this, you have to select minimize all. Minimize all. So it uses universal force field and then it minimizes all the ligands. Then again, you go and click convert all to Autodock, that is PDB-QT. So all the molecules are converted to PDB-QT at once. So in the Autodock, we have done individually. It takes much time. So in Pyrex, it is very fast. Okay. So now we have to load the protein. Okay. So now go to file, load molecule. So in load molecule, you have to load the protein. So in the desktop, we have the Pyrex folder. So there you go there, select the Pyrex, and then you have the protein file. So that is naked protein spdbv 100.pdb. So I will open that. So when it opens, so it will show that file. So this is the protein file. Okay. So now the next step is go and right click and make macromolecule. You have to go Autodock and give make macromolecule. So this becomes the protein. So now you have made the macromolecule. So then you click on Vina wizard. Okay. So here, if you see this, so here it asks click to start. So you have the ligand, you have the protein. So if you click start, so it will ask add the ligands and add the macromolecule. Okay. So macromolecule should be in PDB-QT format and the ligand should be in PDB-QT format. We have all the ligands. We have the protein in the PDB-QT format. So here what you do, You click all the ligands. You can use the shift tab. I'm using that shift and press. So this is the ligand. Then you click the Qt format of the protein also. Then you go forward. So it will generate a grid box. We know the protein molecules. You go to molecules and then you can adjust the grid box. So here these are all XYZs. So you can adjust this. We know the where the XYZ coordinates and what are the receptor residues. So they are it is 20 tryptophan 20. So display label atoms. So here if you see, it will label the display the atoms which you are selected. Then it goes to histidine 110 or 79. Next is 79. So it is tryptophan 79 labeled atoms. So here you can see it has labeled. Then 110, 111, 110, then 111. Display label atoms. Then 122. Phenylalanine display label atoms. So here you can see it is coming outside. So you can drag this. If you scroll this, you can drag this. Then it is 298. So it is 298 display label atoms. Then 300, 309. I'm just giving whatever I remember. So you can adjust the thing based on. So I am adjusting this, adjusting this. Okay. So this also I am adjusting, making sure. Suppose if I am not sure, can adjust this. Okay. So you can adjust slowly. So then you just give forward. Okay. It will start talking. So as soon as you give the coordinates, select the binding spots, it will give the coordinates. So it will first ligand, it starts running. So for every ligand, it will take some time. It will go and start the docking process. Here you can see the completion as soon as it completes the docking process. Similarly, for all ligands, it will complete. So we'll minimize this and I will go to the ligands tab. So here you can see it has created a output for the first ligand. Okay. So these are the ligands. Then in the macromolecules section, if you see, you can see it has created a output file for the ligand. Out.pdbqt is the result file. Okay. So, but this out.pdbqt, it is a merged file of 10 poses, whatever we have seen in the uh, Autodoc. So how to split this? We want to split this uh, 10 poses to single pose. So that is one of the difficulty with pyrx. So you can do it easily. You just watch how it will be done. So this is the naked protein. This is the out.pdbqt. So what you have to do, go to the my computer, then go to C drive, then you go to the program files into 86, then you go to Scripps Research Institute, go to Vina. So you just copy Vina underscore split.exe and you paste here where you are working. So Vina dot split.exe. So now you right click, then open terminal. Okay, when you open the terminal, so it will show. So we will run the CMD. So here another ligand has run. That is why it is showing this. Okay. Suppose you can go and to run CMD. Select. So in the go to the start button, right click and run. You give CMD. You just give CMD. So it will open this terminal. So when this terminal opens, you have to give the directory name. So you have to give the directory name. You give cd gap. You check the path name of this. So this path name you have to give. Okay. So this change the drive to this. It will open that path. So it has entered into this. So now you give uh, vina split. If you press the tab, it will show the thing. So you give just double space input single space and you press Y because we have that split. Okay, then you press tab. Okay, so you are going to split that ligand. So when you press enter, okay, so now you can see it has split. So go to command cmd, give the path name, then you give open the split, then double hyphen input which ligand you are going to split. So when you give this, it has split the 10 poses. Okay. So now we have to create the complex file. So complex file, it is easy to prepare in Autodoc, but in pyrx you have to use some techniques. So I am selecting this pose and selecting the protein. So control, select both the things. Now you go to a program called pymol. So you open pymol, which is already installed in your system, pymol app. So when you open that, so you open that, you can keep nearby. You just drag both the things. So when you open the both the things, you can see the ligand and the protein. So now you go to file, export molecule. So export molecule, then you give save. So you have to save in PDB format. So I am giving as complex dot PDB. You just give save. Okay. So when it gets saved, you can close this pop-up. So now you can see you have saved the complex file. So you can see the other ligands how it is getting finished. Every ligand you have to wait and it gets completed. You split it and you see the interactions. So you have to note down the energy. You have to note down the interactions. So based on that, you will be selecting the top ligands for further experimental validations. Okay. In the Autodoc video itself, I have shown how to visualize the results of the complex dot PDB. So complex means complex of a protein and the ligand. So it is a basic information of the pyrx. Okay. So I hope you will understand the pyrx. You can run the multiple ligands docking using pyrx. Uh, okay. So thank you.